way I organized this talk today was if we were all in person today, everybody would have access to a microscope. They'd be able to look at diseases and insects under the microscopes themselves. So I tried to format this as close to that as I could. I included videos and pictures of things under the microscope, and I just did a little bit of background on plant pathology in the beginning. So I'll just start out. So you can't talk about plant disease without knowing what a plant disease is. So the definition of a plant disease is any disturbance that prevents the normal growth and development of a plant and reduces its value. 80% of plant diseases are caused by fungi. The majority of the plant samples I get in either have abiotic causes or fungal infections. It's not to say that other diseases aren't important. Fungi are just the most prevalent, followed by bacteria, viruses, nematodes, phytoplasmas, parasitic plants, and I bolded abiotic factors because we don't always think about these when we talk about plant disease, but they're so important to the health of a plant. So there are three things you need to get a disease: a susceptible host, a favorable environment, and the pathogen. And if you have all three of these things for enough time, then epidemics can happen. If you break one of these, to control plant diseases, you want to break one of these connections. And one of the most successful things to do to break a plant disease triangle is to plant a resistant host. That's a lot of the time, that's what we suggest if you have a plant disease problem, because we can't control the environment, but we can control what we put in the ground. So I'm not going to talk too much about abiotic factors in the rest of my talk because I want to get into the nitty-gritty of the lab details of plant disease diagnostics, but I did want to mention them. So when you have a sick plant, you need to look at everything that's going on. And this is not a comprehensive list, but it's a list of things that we often see that are going on. Drought stress, excess water, temperature extremes, soil structure, meaning drainage or how hard the soil is, soil pH, fertility issues, deficiencies or excesses, herbicide damage, mechanical injury, as you can see in this image. Not only is this plant very close to what looks like a parking lot and a sidewalk, it has lawnmower injury on the side of it. Wind and storms also cause mechanical damage. Girdling, if you plant a tree and you leave the wires on, that can girdle a tree, or if you don't successfully separate the roots and the root ball, the roots can actually girdle the bottom of the tree. And animal damage, moles and voles can cause significant damage to plant roots. So all of these can be a problem on their own, but generally when I see diseased plants, it's because they've been predisposed, they're already stressed due to one or more of these factors. So abiotic issues often go hand in hand with not only disease issues, but insect issues as well. So the process of diagnostics, what I do when I get in a sample, the first thing I do is read the submission form thoroughly, and kind of tongue-in-cheek here, that's why it's important to fill it out. But I really do read it, and it helps me figure out a lot of times if there are any abiotic factors going on, along with any disease issues. I study the symptoms and signs on the plant, and I'll go into that more later. I look for any background information, any patterns in the fields or in the plant, site, history of the site. I consult reference sources, what diseases the certain host is susceptible to. I consider all possible causes, and then I take the plant under my dissecting microscope and look at it. And if I need to, I'll take tissue pieces and look at them under a compound microscope. I'll mount slides of areas of sporulation or insects or bacteria. I'll narrow down possibilities, rule things out. For some diseases, we have specific tests for exactly that disease, which I'll get into a little bit later. And sometimes if I can't figure it out just using microscopy, I'll cut tissue out and actually plate it onto a petri dish, let it grow out, and identify it that way. And that's called culturing. It's important to distinguish in plant pathology symptoms versus signs. Symptoms are the first thing you're going to see if you have a plant problem. It's an indication of disease by a reaction of the host. So what you notice, like my plant's wilting, I have a leaf spot, a canker, the leaves are small, they're yellow, chlorotic, the roots could be dark. So these are the symptoms. This is a picture of fescue with Rhizoctonia root rot, and those are the symptoms, the dark roots. And signs are different. They're an indication of the disease from direct observation of the pathogen or its parts. This is what I look for when I get a sample in. The symptoms help me figure out what signs I should be looking for. So these are fungal fruiting bodies, mycelium and rhizomorphs, which are, if a fungus is likened to a plant, the mycelium and rhizomorphs would be like the roots. It's like the spreading structures. And I'll also look for a bacterial streaming, which I'll show later. So this is a tree right outside of Townsend Hall. And can you guys see my mouse? I hope you can. These here are actually the fungal structures, these black things here, and that's a sign of the fungus. Other canker diseases might not actually show this, but this is a sign. That's the fungus growing there. All these little black dots within this leaf spot are signs. These are fungal fruiting bodies full of spores. This is Armillaria root rot. It's a wood rot fungus. These are rhizomorphs, and this is a sign of the fungus. It's really helpful when I see these. It aids in diagnostics. So another thing, probably one of the most important things that I look at every single day are spores. Spores are a reproductive structure of fungi and some other organisms containing one or more cells. The first thing I do, well, one of the first things I do if I get a fungal, what I suspect to be a fungal disease, is I look for fruiting structures that contain spores. They can come in so many different shapes and sizes, and when you look at the literature, it's really easy to tie them to a certain disease. These spores here are Alternaria. This is the shot hole fungus, Blumeriella, and this one on the right side is Botrosphaeria spores. Another thing that's really important to look at in diagnostics are patterns, which is why I love to get pictures with samples. An email with a picture can be so helpful. Looking at the percent of the crop that's affected, or even the percent of the plant. So I look at patterns within a whole planting and also on a plant, what the pattern is. A lot of leaf spot diseases start on the bottom leaves of plants and work their way up. So if you submit a sample to me that says only the leaves on the top are affected, then that makes me think in a different direction. It's not ruling out that it could be a pathogen, but it might make me think it might be something abiotic. So patterns are always important to have that in the back of your mind. The site is also very important. Is it a low area? Is it in the shade or the sun? How's the drainage? 9 times out of 10, if I get a plant in that has a root rot, it's in a spot with poor drainage. And time is really important. So if symptoms appear very quickly, like overnight, it's pretty indicative that it's probably not a disease issue. Diseases have an infection period. The pathogens take days and sometimes weeks to progress. Sometimes it will look like a disease happened overnight, but the pathogen was there for a lot longer. It just took the right environment for it to start showing the symptoms. So here on the left is a picture of a disease pattern or distribution, and on the right is an abiotic or not biotic distribution. So on the right side, disease distributions are often random or scattered, or they happen in localized spots. I know this isn't ornamental. This is a watermelon field. You can see here it follows no pattern. It doesn't go along the rows. Doesn't look like it's in a low spot or anything. It's just random within the field, and that's pretty common for diseases. Here on the right, we have a hosta plant that looks kind of random, but this is actually sunburn. You can see the leaves are affected on the edges and the tops here. And what clued me into this being sunburn is this V shape right here was not affected, and was exactly under this leaf. So if you ever have a plant that you think is caused by sun scald or sunburn, look to see if any of the underleaves have been shaded by the ones above them and aren't affected right there. That's pretty diagnostic for sunburn. This is a boxwood shrub that has Phytophthora root rot. Root rot diseases often only affect one side of the plant or just completely random on the plant. This is a disease distribution because it's only on one side of the plant. If you had like a fertility issue or a sunburn or fertilizer drift, or herbicide drift, a lot of times that would happen only on the top part of the plant or the bottom of the plant, not like one-sided like this. This is a fur sample that we got that had necrotic tips only on last year's growth. So this year's growth right here is healthy, and it was only on the tips here. So I looked at it under the microscope. I found no fungal fruiting bodies, and that, along with the pattern here, clued me in that it probably happened last summer. Maybe this plant dried out, it didn't get enough water, it scorched these tips, but the new growth is okay. And if this was a disease, it would be on the new growth, and it would be more random on the needles like this, or not like this. This is another example of an abiotic issue. This is maple leaf scorch. Oftentimes, if a plant's been scorched, it's hot, dry weather, or hot, windy weather. The transpiration out of the leaves happens quicker than the plant can bring water back up from its roots, and it scorches the edges of the leaves. This one's kind of tricky because maple has a bacterial disease that can mimic this, but there are other ways to test for bacterial diseases. But if you see necrotic tissue just on the edges like this, it's probably leaf scorch. So this sample was pretty neat. There's obviously fungi here, but it's not causing the symptoms on this plant. This was, I think it was a boxwood plant that I diagnosed botrytis, which is a fungus, and it is a plant pathogen, but it's not a pathogen on boxwood. So it's not causing any disease on this plant. And then I looked at the pattern, the distribution here. You can see right here, it's just on the tips of the leaves. So my theory is that these got scorched somehow. They were in, I think, a box store, and they got scorched, and the tissue died, and the Botrytis came in, or botrytis came in secondarily, and is just on that necrotic tissue. That's something that I see a lot, secondary organisms, and sometimes it's really hard to define what is the main pathogen and what came in secondarily. Here's another pattern on a tomato plant. This turned out to be herbicide damage, but this sample was tricky because these symptoms can also be viral. So tomatoes get a number of virus diseases that can look like this. I tested for a number of viruses, it was negative. Brian and I checked for broad mites, which can also cause symptoms close to this. Didn't have them. It turned out that the manure that this person was using in their garden had herbicide residue on it, and that was causing these symptoms. And here's another picture of leaf scorch. And this was only on one side of the plant. And I think it was just on this one side because you can see there's a road right here. The other side wasn't showing any of these symptoms. So I think this happened on a hot day, and this side of the plant just didn't have enough water because the road was right here, and it got scorched. So the other thing you have to be cognizant of when you see a diseased plant is the site. So this tree looks very bad. If I got a picture like this, I wouldn't really know where to begin. But if you look, this is the base of it. That's obviously what's going on. It's sandwiched between a building and a sidewalk. It has a grate right in the middle of its root system. So it's important that when you see a diseased plant, you look at everything that's going on to try to figure out what's wrong. Okay, so that was kind of the introduction. And now I'm just gonna show you a couple samples that I got in over the past month or so and how I diagnosed them. So the most common root rot pathogens are Phytophthora and Pythium, which are oomycetes. That's a fungal-like organism. And then there's Rhizoctonia and Thielaviopsis, or black root rot, which are true
fungi. Jill, before you talk about the root rots, we had a question from Natasha, wanted to know how you can know if the manure has herbicide in it.
I honestly don't know. Before you plant, I don't think there's really any way to know. There might be labs that do testing, but I don't know off the top of my head.
Yeah, probably just understanding how it's been managed.
If any of you have heard of black root rot, it's a fungal pathogen that attacks plant roots, and it has a very large host range. I'm not going to mention them all, but some of them are on the left-hand side. And one of the susceptible hosts is carrots. So this is a really fun activity that you can honestly do at home. If you suspect you have a plant that has black root rot, what I did was, you can see the roots right here, and I surface sterilized them in 10% bleach and water. And I also surface sterilized the carrots. This was carrots from my house. I didn't have any in the lab. And then you sandwich the roots within the carrots, put them in a Tupperware with a moist paper towel, let them sit for a week or two. And if the fungus is there, it will infect the carrots, and you can obviously tell. And so those are the symptoms. And then these are the signs of the disease. I honestly think that you could see these with a hand lens. These are the biggest spores I've ever seen. And here's a picture under the microscope of these spores. This is a Thielaviopsis spore. They look like caterpillars, and they're very dark. So the roots in black root rot are actually turned black because there's so many spores within the roots, and they're dark color. So that's where it gets its name. Another root rot pathogen that you may have heard of is prolific Phytophthora root rot. Has a ridiculously large host range. I've put some of the hosts to the left here, but there are more than that. I previously showed a picture of a plant dying on one side. If you have plants in like a low area or area with low drainage and they die, it's likely that Phytophthora may have been involved. So if you dig up your plant and you see roots that are dark, their cortex pulls off easily, or even the roots break off easily, that's an indication. Those are symptoms of a root pathogen. And this is just a picture of Rhizoctonia root rot, but it looks similar to Phytophthora. So I'm lucky enough to have these serological tests, which are lateral flow tests for this disease. It tells me within 30 minutes if the disease is there. So it's this packet here with buffer inside. You smush the tissue, a very small amount of tissue inside, and then the buffer binds with proteins on the pathogen, and then it wicks up on this diagnostic strip, and 2 lines mean positive and 1 line means negative. So it's really easy to tell if there's a Phytophthora infection. However, it only diagnoses to the species level. There are probably over 100 species of Phytophthora. So if they're for like research, or if you wanted to know specifically what species is there, this isn't adequate enough. So in that case, you can plate it out on media. So this is Phytophthora growing on a specialized media in a petri dish. You can see the fluffy mycelium here, which is basically like the tendrils of the fungus. And here's a picture picture of Phytophthora mycelium under the microscope. Phytophthora and Pythium are oomycetes, so they have very distinct mycelium, and I'll try to walk you through it. There are actually two, there's Phytophthora here and another unknown fungus. So if you follow my mouse right now, this is the other. Okay, I'm getting a notification that says my internet is unstable. I hope that I don't break out. Okay, I'm gonna keep talking. So if you follow my mouse here, this is not Phytophthora, this is a different fungus, and you know that because it has these tiny little cross walls here. Almost all fungi have these on their mycelium, and Phytophthora and Pythium don't, so they have very distinct mycelium. So if you follow my mouse now, this is part of the Phytophthora mycelium, and you can see no breaks in it. And so this here is an oospore. This is the resting structure of Phytophthora. So this is the dangerous part that overwinters in the soil and can infect plants next spring. I know it doesn't look like much here, but this is their overwintering structure. And here's another one right here and another one right here. So now I have a video about bacterial leaf spot ID. So bacterial leaf spots, if you're unsure if you have fungal or bacterial leaf spots, check if they look more circular or angular. If they're very angular in appearance, they're probably bacterial. And I look for bacterial streaming under a microscope, and watch the video. So I'm going to show how I diagnose the difference between a fungal leaf spot and a bacterial leaf spot. So I have 3 different leaves here that I think might be affected by bacterial leaf spots. This is a black-eyed Susan, a cherry, which has shot holes in it, which can be caused by a fungus or a bacteria. And this is oak leaf hydrangea. A lot of times you'll be able to differentiate a bacterial leaf spot because the spots look like they're delineated by the veins. They can be angular. And so what I do for samples like this is I cut up the leaf spots with a sterilized razor blade. So here I'm just cutting out one leaf spot. I went right through the middle. You should have and then I put that piece I cut out that has some of the lesion in it on a microscope slide, which already has a drop of water right here. And cut out a few pieces. And then once they're in that water droplet, then I just cut them into a lot more pieces, tiny little slivers. And so what this does is if there are bacteria in that leaf spot, they'll stream out of the tissue into the water droplet, and I'll be able to see it under the microscope. So I did the oak leaf hydrangea lesions under the microscope, and they were streaming. So these are bacterial leaf spots, which makes sense because they're angular. They don't have a lighter spot in the middle, which is more indicative of a fungal infection. So now I'm going to look at the oak leaf under the microscope, or not oak, sorry, cherry. All these shot holes, and I already cut out a piece. So I cut out a piece here that still has some of the tissue intact within the lesion. And I'm just going to chalk that up in a drop of water on my microscope slide, just like I did for the oak leaf hydrangea. I already have a drop of water. Cut it into tiny pieces. Get a lot of surface area, so if there is bacteria there, it has a bunch of opportunities to stream out. And then I'll put a glass cover slip on and look at it. Spoiler alert, the cherry was not bacterial. I'll show that in a minute. But this is bacterial streaming. It is not always this distinct. I actually kind of cheated here. This is a corn sample. The oak leaf hydrangea did stream, but it wasn't as photogenic as this. This is pretty much as good as it gets. What this is actually the bacterial spores streaming out of the plant tissue. So over here on the left side is the plant tissue, And all of these are the bacterial spores just streaming out. And this was my microscope's highest magnification, so there's no way to actually identify the bacteria here. They're way too tiny, but it's diagnostic for a bacterial disease. And here's another video of also of a corn sample of bacterial disease. So it's coming down this way. And that's more like what we see. It's more like molasses or like a glacier paste. It's not always like the last video, but it's really, it can be very distinct, and when I see it, it makes me happy because it's an easy diagnosis. So like I said, it's impossible to identify bacteria to species or genera just from streaming. So if I want to know exactly what bacteria it is, I'll plate it out on a Petri dish. So what I do is I'll cut up the pieces just like I did in the video and let it sit for an hour or half an hour in a Petri dish with water. And then I'll take a bacterial loop, which is just a wire loop, and stick it in the water and then streak it out on the Petri dish. And then within a day or two, the bacteria will grow. And each one of these dots is a single bacterial colony. So that's a way to get a pure culture. And then from there, I can do other tests based on the color. If it fluoresces, if the bacteria is gram-positive or negative, I'll be able to tell what it is.
Jill, we have a question whether or not you sterilize the blade or use a new blade when you're working with the leaf samples.
Yes. Yep, I flame sterilize between them. Okay, so like I said before, the cherry ended up not streaming. So I looked under the microscope again for if there were any fungal fruiting bodies within the spots, and I did find a couple. And I don't have a picture of the fruiting bodies, but I have the picture of the spores right here. And it's kind of hard to see, this isn't a great photo, but these, a spore is long and skinny with one septa in the middle, so it has two cells in these spores. So this is a shot hole on cherry caused by the fungus Blue Muriella. This is a horse chestnut, no, not a horse chestnut, a white hazel sample with fungal leaf spots. And this isn't the greatest video, but I tried to film what I do when I'm under the dissecting microscope looking for fruiting bodies. So fruiting body is basically the structure of a fungus that holds all the spores. And spores are basically like if a fungus is a plant, the spores are the seeds. That's what's going to disperse in water droplets and wind. So what I was trying to zoom in on here is, so all of these things here, all these yellow things, this is perfectly normal for this plant. These are just like the leaf hairs or trichomes. What is abnormal are the black fruiting bodies. Like one is right here. There's one right here. And I think what I was trying to zoom in on is a fruiting body here that actually has spores coming out of it, the white right there. And then this is what I do so my dissecting scope only goes up to a certain magnification, and to identify spores, I have to put them on a microscope slide under a different microscope. So this is what I do to actually get the fruiting bodies. So there's one right here, and there's one right here. So I have to have a very steady hand and actually pick out those fruiting bodies, put them on a microscope slide to identify. And so this is what I saw when I looked under the compound microscope. So these star-shaped things here are those leaf hairs that we saw before. These are just like the trichomes, perfectly normal. This orange stuff here is the plant tissue. And then this black spot here is the fruiting body. With that hole in the middle is where the spores come out. And then all of these little egg-shaped things are the spores. So on this witch hazel sample, this ended up being Philosticta, and these are Philosticta spores. I think I went in closer. And that is the same disease that causes these leaf spots on maple. Here is this is a little bit overexposed, but here's the leaf spot on maple, and all these black spots right here with the white coming out, these are the fruiting bodies, the pycnidia of the fungus, and the white is actually the spores coming out. There are the spores. So this is a video I took of a different disease, and these are the spores streaming out of the fruiting body once they were put in water. So I believe what happens is a lot of fungi, once their fruiting bodies get wet, the spores expand inside and there's no more room within the fruiting body. So they expel the spores. And it's just amazing how this is thousands of spores right here from one fruiting body in one leaf spot on one leaf of the plant. So you can just have an idea of how many spores are out there for diseases. Not all of these are going to cause disease though. Another thing I just wanted to mention briefly are rust diseases. So rust diseases are kind of like powdery mildew in that if you see it a couple times, you can become really good at diagnosing it. Because they have bright orange spores. This is the only fungal pathogen I know that has bright orange spores. So if you think you see a rust pathogen, they sporulate on the underside of the leaf. So flip the leaf over, and if you rub it with your finger, sometimes you'll actually see orange on your finger, or if you rub it on a piece of white paper, you'll be able to see the orange spores. And this is a little video of Brian, correct me if I'm wrong, it was a midge larva or a fly larva crawling around on a sample with rust, and it's just covered in rust spores all over its head. I'm going to play that again. I just thought that was interesting, spreading it around that leaf. And the neat thing about rust spores is they're actually orange. So when you look at them under the microscope, they're very distinct. Another common thing that I've been seeing are Botrisphaera cankers on woody ornamentals. So this was a maple that had, this was the first symptom that I saw, this dark discoloration here. And I wetted the sample here just to make it more pronounced. And then on the right side here is when I cut into it, so you can see the vascular discoloration there, cutting off the vascular system. So the sample actually was profusely sporulating. So what I did was I cut a little piece off right here in the middle of the canker, and I put it under the microscope, and I put a dot of water on it. So all of these are, the black parts are the fruiting bodies, the pycnidia, and the white stuff coming out of all of them are the spores. So once I put that drop of water on, the spores just kept coming out. And then this is a picture of them under the compound microscope. So the fruiting bodies combined with the spores and the symptoms on the plant, I was able to identify them as Botrisphaera. So all of those things together. This one I haven't figured out yet. I just saw it this morning and I thought it was neat and I wanted to show you. This was the same cherry tree that had the shot hole disease. On the other side of it, it had these signs of a fungus. At first I thought it was hypoxylon canker, which is common on oak trees and looks similar, but cherries aren't a host of that disease. So I actually emailed Nancy, and she wasn't sure either right off the top of her head, but she thought it might be a Cytospor canker, which is a different fungus. So what I did is I took a piece of that black tissue there, which was the fungal tissue, and you can see where my mouse is pointing. These are masses of spores. And I put those under the compound microscope, and you can see all these neat spores. These are ascospores in chains here. So this will be one of my projects tomorrow, to look in the literature and try to figure out what this is, but I thought it was neat. And the last thing I'm going to talk about, taking a lot of time, is rose rosette virus. So the symptoms of this are very distinct. You get leaf reddening, deformation, curling, proliferation of thorns. It almost looks like herbicide damage. So the symptoms of this disease are very distinct, but it's actually hard to diagnose because the only way we can diagnose this is through molecular methods. And our lab isn't set up for molecular methods yet, hopefully within the next year. So the second best thing is looking for the insect vector. So I can never say this, Eriophid mite is the vector for this virus. And if you can find that mite along with these symptoms, it's like 99% sure that's actually what's going on. But the only way to completely verify it would be molecular methods. So the mites like to hang out in the flower buds or leaf buds. And here is a bad picture. This was my highest magnification here of the little mite. And I put it under my compound scope and I smushed it. But these are tiny, tiny little mites. So if you have these very distinct symptoms and you find the mites, it's pretty obvious that you do have rose rosette virus. So that's how I would diagnose this at this time. And that's all I have for today. I can't wait until I can do this in person with people. If you want to find our website, are thinking about submitting samples, just Google UD Plant Clinic. That's the easiest way to get to it. My email is right here. And I also started an Instagram, if anyone's on that, for our diagnostic clinic. And that's all I have. Thank you guys for listening.
Thank you, Jill. That was awesome.
Today, I'll be introducing an extension scholar that I have remotely working with me. Her name is Molly Wyatt, and she's going to be talking to you first. So we're going to go into the virtual hands-on entomological experience. Here I'm, going into my hole, which is my cave in my house. Sometimes it's a lab. Summertime is a time for me to be outside, not indoors. So I've been going stark crazy with this whole pandemic, let's do remote, not go out and play with critters thing. So Molly's going to talk to you a little bit about gloomy scale and then insect diversity. She's going to cover it. This will be one of her first times talking to a group of professionals and master gardeners. So as she makes comments, I may occasionally interject in through her presentation, but I'm going to let her handle most of this, and then I'll jump back in to where I'll talk to you briefly about the exotics that we are currently concerned about, what we're watching for, and then I'll also bring you a closer look into some of the insects that commonly require further microscope work. It's not like the pathology side where you're looking all the time for fungal spores or hyphae or stuff like that for some of your samples. Luckily, our insects are larger, usually, or generally speaking, I guess. The only problem is they also don't like to sit underneath a microscope very often. So that makes it a little bit of a different experience. And then I'll have some observations of things that just are incidental, but you may be seeing at this particular time of year. And with that, I am going to go ahead and follow the rest of you and practice the science of snoozing or napping as Molly Lee takes on her part.
Okay, so the gloomy scale is primarily a pest on red maple, but it's also been observed on sugar maple, elm, tulip poplar, hackberry, and several other species. The female adult covers are called test and can be up to 2 millimeters wide with a central pale ring. And beneath the test is a soft-bodied scale insect, which is usually yellowish, legless, and wingless. The males are smaller, have an oval-shaped armored covering, and develop legs and wings as adults. And the nymphs are called crawlers and are less than 1 millimeter, and they can be orangish to yellow in color. In this first slide, these are the crawlers. So you can see them here. They probably settled about 2 weeks ago, and they are this year's brood. So in this slide, it's a female gloomy scale where the top has been flipped off to reveal the actual scale insect. This female has probably not laid eggs yet or is in the process of doing so. So you can see where like this is like the top of the scale that's been flipped off. Again, this is another view of the female with the top off and others around it from last year's brood, along with the new crawlers that have just established. So these are the newer established ones, and then topped off female, and then other females. This little yellow circle is of the immature scale with its top flipped off. So that little guy right there. So damage caused by, I've been calling them gloomies for short, is caused by their piercing, sucking mouth parts. They penetrate the bark and feed on parenchyma cells, and those cells synthesize and store organic products within the tree. When investigating a tree you should suspect has an infestation, it is extremely important to identify the stage the scale is in. You need to figure out if they, 1, are alive, 2, if there are eggs, 3, there are mature females, and 4, if there are living this year scales. Knowing the stage is very important so we can come up with the best control method. So for example, you may have a tree that is covered in scales still from the past year, but they're dead. So you could treat them, but then the following year, it still looks like the exact same. The scales cement themselves to the tree and don't just fall off when they die. It should be noted it's pretty difficult to flip off the tops of these buggers. So if you are not experienced, always take it to a professional. As well, like in this slide, many heavily infested trees can have multiple species on it. In this slide, the brownish guys, so these little guys here, and the white little slivers are Japanese maple scales. So taking it to a lab, they can identify all of the species and control accordingly. In this slide, we wanted to show that because the obscure scale and gloomy scale are all literally almost identical, the difference is that if a scale is on an oak, 95% of the time it would be an obscure scale. Whereas if it's on a maple, 95% of the time it would be a gloomy. Important to take note of the host plant as well, because crawler species timing is different for different species. In this video, you'll see what happens when a tree is heavily infested. It's evident that there's thinning canopy, twig and branch die back, and it can eventually lead to death. Close up right now shows the black nodules, which are living or established scales. And then near the end of the video, which is coming up, you'll see the white patches. That's from last year where the scale has gone away, but leaves behind a cement-like residue that's used to attach to the tree. So ways of control, again, want to stress how important it is to identify the stage of the scale. When in doubt, again, take it to a professional. There are some biological controls, such as parasitic wasp in the genre Signiphora, Incarcia, and Ablurus. Also lacewings, ladybugs, and predaceous midges. But in an urban landscape, you need to have a diversity of plants, a wide diversity, to help with temperature, alternative resources, and vegetation refuge. But we'll talk more about plant diversity in a little bit. Oh, the last control I'll mention is horticultural oils. So the oil, you'll use 1 to 2% when crawlers are active, no more than 2% during summer, and then 3 to 4% oil when the tree is actually dormant. And that's all I have for the gloomies. And then Brian, on to you with the galls.
This is a far out view of the bald cypress. Leave it here for a second. Bald cypress twig gall midge. This is a nuisance pest that people frequently are concerned about because populations can get pretty high. Go ahead and forward it. Here we see a close-up of the gall on the left. On the right, you'll see these thin little white strips. I don't know if you can see my cursor, but to the right, the little white strips that are on the main stem, yep, those are shed skins of the bald cypress rust mite. It's an aerophyid mite. So it's similar, it's closely related, same group of mites that Jill talked about with the rose rosette. This one just happens to be on bald cypress, and it'll cause a bronzing of foliage in late July through mid-August, especially if it's really dry out. To the left of the or just above, keep going up. Yep, there you go. That's actually a picture of the aerophyid mite. There might be another one on another slide that's a little bit closer. You can go ahead and forward and you'll see. Yeah, so this is a little darker, but the aerophytes are carrot shaped in their appearance. And they have 4 legs at the head end. Your spider mites, such as the 2-spotted spider mite or the spruce spider mite, will have 4 pairs of legs. Aerophytes have 2 pairs. So they're different types of mites. And unfortunately, miticides for aerophyid mite control are limited. Aerophytes are difficult to target, and the products generally don't work as well against them as they do against the more common 2-spotted spider mites. Okay, if we want to go ahead and forward through it. So when the sample comes in with the twig gall midge, basically I set it up under a dissecting scope, take a spatula or a razor blade like she's pointing to, and begin to carefully cut into it. Eventually you'll find the pocket. This pin is basically pointing just to the right, is a small hole. That's where the insect feeds, and it'll eventually form a little chamber, which the pin is pointing to, that it'll pupate in. And okay, next. There you can pull out the little culprit that's responsible, and it's hard to tell what it is in this picture, but it gives you a sort of size reference to the size of the gall that it was in. Here you can kind of make out at the bottom of this brown the reddish colored eyes. It is a fly that's essentially a little bit bigger than a fruit fly, but this is the fly's pupa case, and it's just about to come out. Here it is set out on a petri dish, so you can see the pupa as a whole, and there you can see the eye that she's pointing to. The twig gall midge rarely does any long-term health impact to the bald cypress. It just tends to make the tree look a little unsightly. Management would be to rake up the needles that fall from the tree in the fall and move them away from the tree.
Having a wide diversity of plants is so important when it comes to pest management. By increasing plant diversity, you increase the number of insects, which in turn decreases pest pressure. When we showed the video before of the gloomies of the tree, did everyone notice that there was like nothing around it? Well, when you isolate a plant and have no diversity around it, you are basically giving that pest like a monocultural habitat. It's like an all-you-can-eat buffet with no repercussions. So for a natural enemy to get to that pest, it ends up too costly with the handling time. Yes, eventually a natural enemy may show up, but usually after pest populations build to high levels, which in turn makes it hard to reduce the pest populations to acceptable levels by beneficial arthropods alone. So when you have increased arthropod populations due to diversity, they may not all be beneficial, but are just incidental. If you have an abundance, then those natural enemies of pest won't leave that area, even if they eradicate the pest. They will still stick around, feeding on those incidental arthropods. Then if pests increase again, those natural enemies will zone in on them. So the next few slides are of spiders. I know everyone's favorites, but realistically, they are extremely important predators. So in this photo, it's of a dark fishing spider feasting on a spotted lanternfly nymph. The next slide, I'm not sure what species spider this is, but it's feeding on a brown marmorated stink bug. They feed on many different pest species, so you want to have a lot of diversity to keep them from leaving the area. The last spider is the yellow garden orb weaver. These guys are just amazingly beautiful and will eat a lot of pests as well. Oh, and I forgot an adorable jumping spider because they're just adorable. A few other photos coming up is just of things found in a diverse garden. So these are some hoverflies, which are beneficial. We have damselfly and ebony jewelwing, the red admiral skipper, which is beneficial, leafhopper, and an Argus tortoise beetle. This one is a unicorn caterpillar, and this one I am not 100% sure what that caterpillar is, but all caterpillars are beneficial. Like Thielaviopsis always says, they feed, I forget the exact percentage, but a huge percentage of birds. And then lastly, I have an eyed click beetle and then another form of a click beetle. And like this eyed click beetle feeds on small arthropods in the soil, and it also feeds on pollen. So when it goes like plant to plant, it's an accidental pollinator.
One of the important things that I wanted to share with you to be aware of is that there is a new exotic invasive pest that has been found in Sussex County. It is the Crape myrtle bark scale. Crape myrtles have generally been relatively pest-free, but so crape myrtles have had aphids that'll feed on them. They can be a nuisance for that, and the fact that they produce honeydew, the sooty mold gets on it and looks a little darkened. You'll have Japanese beetles that may occasionally feed on crape myrtles, but by and large haven't had that huge an impact on the health of crape myrtles. Crape myrtle bark scale, as you can see, can be pretty extensive on crape myrtles if left unchecked. It came over from Asia. It has been found in the southeast, and it's thought that the sample that was found in the landscape in Sussex County came from a plant that had been purchased recently and installed. When you flip the covers over, you've got a reddish to salmon colored insect underneath this very white waxy substance. We typically call scales armored scales or soft scales. That is the oversimplification of the scales that we can encounter. Their diaspidids are the armored, the coccidae are the soft scales. There are felt scales as well, and this is it's a separate family of scales, but they're all in that true bug group. And so this is a felt scale. So the biology doesn't quite match up to the armored scale or the soft scale. They are honeydew producers, and it can be quite extreme. And so then you'll have a lot of sooty mold showing up on your crape myrtles, which can limit photosynthesis of the plant if it becomes extensive. In the South, we do not know how many generations we're going to have. They also produce hundreds of eggs. We don't know how many generations we're going to have here in Delaware. The thought is anywhere between 2 to 4 generations in the Southeast or the Gulf Coast states, or South Carolina potentially, 2 to 4 generations. For us, probably closer to just 2 if I had to guess. But again, it's hard to say. There's a lot of the biology of this pest we just don't know yet. So basically, if a sample comes in, I throw it under a scope, and this is a close-up that Tracy and I took of that sample that was taken in Sussex County. Looked at it under a microscope, and basically this pointer that you can see here is pointing to one of those felt coverings over the scale. This was a really old sample, so there weren't any living insects underneath. Here you can see the remains of one. It's dried out in kind of a reddish brown. This one, when I flipped it over, and it will flip over like any of the other scale coverings, revealing that reddish colored or salmonish pinkish colored insect underneath when it's alive. There was nothing under this one. Initial populations that hit crepe myrtles are hard to find, so you have to be diligent in your observations on your plants. If you're scouting, just look closely for increased amounts of sooty mold or honeydew. And if you see sooty mold or honeydew on your crepe myrtles, take a closer look, because it could be something else other than aphids. The crepe myrtle bark scale, the name is descriptive in the fact that the scale is typically going to be found on the bark, not the leaves. And it's going to be on branches and trunk.
Brian, we have a question if you can use warm soapy water with a brush to remove the scale.
I would say you'd need to be very careful about using soapy water for any plant, because I'm assuming they're talking about soapy water from soap in their house, like dish detergent soap. I would not use dish detergent soap on a plant because you run the risk of severely burning foliage. For the trunk or the bark on the crepe myrtle, it wouldn't be as big a deal unless you got plants around it. Dish soap is not meant to be used on plants. They're meant to be used on dishes. If you want to use, and when you hear me mention insecticidal soap, it's a different type of soap. The carbon chains making up that soap are different. I believe there's fewer carbons associated with it, which means it's going to have a less likelihood chance of burning foliage or harming the plant, but it still has the impact on the insect. So it's been tested and has been shown to be safer to use on plants than dish soap. That being said, insecticidal soap, if you're not careful with it, will burn the hell out of your plants as well. So you just have to use sound judgment. If you use dish soap, just make sure you're not being willy-nilly with it and hitting collateral plants that you don't want to burn foliage on. You could potentially remove some of that with a nice stiff wire brush. It would be an option to use, but it's going to be labor intensive versus frequently used insecticides. The problem is the insecticides that work best is still being worked out or determined. One that is known to have success are the neonicotinoids. However, with current concerns about potential impacts on pollinators, you'd want to be using those, if you do, after the plant has flowered. And so depending on how many generations, I mean, you would still, I would suspect, get management. If you applied it in late August, you'd probably have some impact, and the plant would have already have flowered by late August, more than likely. So your impacts on pollinators would be less. But that would be something you'd have to be cautious about. The other thing you can use horticultural oil at the dormant rate that Molly mentioned earlier, the 3 to 4%. When the crepe myrtle's gone dormant, you could do bark sprays with that, and you would get some level of reduction. How much? That information in terms of how efficacious that is still being worked out. You could also do it in the spring before the plant leafs out. You could follow it up in the spring with another application of horticultural oil to have some efficacy there as well. If you are a professional, there are insect growth regulators that you could apply talus or distance that would have impact at reducing their populations, and you may get really good control with them. You just need to target the crawler stage. One of the challenges is that it has an overlap of crawlers and adults at any given time. So you're going to have an overlap of generations at some point. The other insect, I noticed the chat thing is flaring a fair bit. I guess somebody will hit me up with questions here in a moment. Spotted lanternfly, I wanted to briefly mention it. I'm not bringing samples in. This insect is plenty large enough and distinctive enough to identify without having to bring it into a lab and put it under a scope. The reason why I'm mentioning it is we are, if you look at these two pictures, one on the left is spotted lanternflies in the first, second, or third instars. We're at the stage where you're looking at the one on the right, the one with the red insect. That is also a spotted lanternfly in its last nymphal stage. Right now we're having a mix between the black and white polka dotted one and the red, black, and white polka dotted insect. It means we're about to have adults. It won't be long before we're having adult spotted lanternflies. And the quarantine that Newcastle County is under means that our vigilance for watching for adult spotted lanternflies needs to go up. Otherwise, the adults are fantastic hitchhikers, and we don't need to take them to the beach with us over the weekend. So before we leave to go sightseeing anywhere, whether it's vacation or work, as time permits, quickly look in wheel wells under and bumpers of cars so that we're reducing that opportunity for adults to further disperse throughout the state. Everybody does their little bit here and there, it'll help. All right, so that's spotted lanternfly. Here's a close-up of the nymph that is right before the adult stage. And then to the right, we have an image of what the adults look like when their wings are folded. When they open them up, they actually have a nice bright red spot. They are a hopper. They are not a moth or a butterfly. They're in that true bug group. They typically feed through the trunk of the tree or through branches. On the right, we have a picture of their egg masses. Hundreds to thousands of adults are possible, and although there can be lots that we're seeing this year, don't worry if you haven't seen them yet, you will before long. Their populations are only going to go up from here on out. Luckily, there are a number of management options for those. Unfortunately, most of those involve neonicotinoids that have the best efficacy for any length of time. They'll lay their eggs most anywhere. So on your car, on your lawn furniture, under your deck, on rocks, picnic tables, you name it. If it's a nice hard surface, they're going to lay their eggs there if they get the opportunity to this coming fall. Here's pictures of egg masses on rocks, picnic benches, and so forth. So that was the exotics in 6.3 seconds. IPM is an incredibly important part of insect pest management. Being vigilant, scouting, and monitoring are the biggest pieces. Making sure we're observant of where we're looking. And then the identification is important so that we're sure we're putting products that do the best job for the pest in question. And then, of course, there's cultural practices, biological and chemical control, and keeping records of what we do. I'm going to try to move quickly because I only have about 15 minutes for you to enjoy the excitement and wonders of insects. This is an image of fresh toothpicks that are on a cherry. This is a sign of ambrosia beetles or bark beetles. To get an idea of which species it is, I'll frequently have either homeowners or professionals send in samples. Tracy gave me one to take back up here to investigate in the lab. She sent me pictures where I got to see like the frass toothpick coming out of the tree. I didn't get to see the sawdust that comes and falls around the base of the trees. The sample I got and looked at in the lab, I was banging around in Jill's lab this afternoon, trying to get a nice image to show you. What I got to see from the sample Tracy gave me here recently, I did not get to see the nice staining of the wood from the fungus that the beetles actually eat. The fungus jams up the conductive tissues of the plant and it ends up dying that way. So basically I pulled off some of the bark, and in fact there were a number of emergence holes from the bark beetle. So then basically I work at trying to see if I can get the insects out so I have an arthropod to identify. The arthropods, or the insects that do this damage or boring into this tree, are about the size of a grain of rice. So they're small. So basically I did a cross section, which we see here, and you can kind of see the beginnings of that fungal staining in the conductive tissue. And here you can see the gallery that the larvae did as they went feeding through the stem of the tree. I did find a dead insect stuck in one of the emergence holes, but he had broken into pieces, so I wasn't able to identify it. What we would like to see is either the larvae, so that we know we're getting that portion of that population under control, or some of the eggs before they start actually damaging the plant. Two of the common species of bark beetles that we have attacking plants in this area are the granulate ambrosia beetle and the black twig beetle, or Xylosandrus germanus. Again, these are highly magnified. Imagine these being the size of a grain of rice chewing into the trunk of a tree or a branch of a tree. They typically will go towards the trunks of trees, and usually young establishing trees. They will go after trees that appear to be healthy, but those trees may still be suffering. And trees in our landscapes the past couple years have been pushed and pulled in terms of their health due to drought or excessive rains. And so right now, even mature trees are going to be susceptible to these particular insects because they're dealing with environmental factors that are making it hard for them to get by. And so too much rain, like we had last spring, stresses it out. To follow it up by drought, those trees are still dealing with the drought that they suffered with last summer. And that stresses them out enough to give these insects a chance to get in. Bark sprays of bifenthrin or permethrin are your best options for dealing with these. You can see the lists of host plants for both these species here. It's wide. Luckily, mature trees in the landscape have a better chance dealing with these and handle it a little bit better. They can take a year, maybe two, before succumbing. Young trees in a nursery or recently planted trees are probably going to die and need to be killed. Bagworms. Most people are familiar with the bagworms we see on the left hanging in junipers, yew trees, arborvitae. One of the things that's nice, and Tracy and I mentioned from time to time, is looking for the eggs that are hatching. So if you pull bags off from September until May, as you get into May, you can open it up, and Tracy took this picture. You can get in and see that where these arrows are pointing, those are what we call neonate caterpillars, or first instar caterpillars. So they hatch inside the bags, and then they crawl to the bottom and throw out silk and blow on the wind to nearby plants. Now, it may be the same plant that their mother was on, and they'll feed there, but they'll throw out silk to disperse to other plants as well. Depending on how much dispersal goes on, helps determine on how much you need to focus your management applications at the beginning of June through the end of June. After they settle, they feed and hold their bags upright like we see here. This is called the dunce cap stage, and it's the best time to do your management. It's usually before the end of June or the first week of July when they're in this stage. And you can make an application of BT and get excellent control. There are other products available for bagworms as you move throughout the year. Here's a nice brief video of the bald cypress twig dull midge. In this shot, before I start it, you will see The cone of the cypress, it's often confused as being this gall midge, and it is not. So this is the first part, that's the cone. And then here you have the gall midges that I'm turning over so that you can see the foliage growing to it. The insect is inside there, and eventually as it matures, it chews its way out and will lay eggs the following year. Armored scale differences. Soft scales do not have a test. Armored scales do. The test is that scale that can flip up and still reveal the insect underneath it. Soft scales, if you flip it up or flip it over, you actually end up removing the whole thing. Armored scales feed on the parenchyma tissue, as Molly had mentioned. They produce far fewer eggs, and they do not produce honeydew because they're feeding on that parenchyma tissue. They're not feeding in that liquid diet that the soft scales do. Jill and I took this picture of Crape Myrtle or Cryptomeria scale. This was on a Canaan fir. Here you see the insect here. This is the cover. You flip it off and the insect is revealed underneath. Here you can see also where another insect has been flipped off. Covering over another mature adult. They often will look like fried eggs. They'll cause banding on the host plants that they're feeding on. This has 2 generations a year. One of them is earlier in the year, the next one usually around June. The second generation crawlers are coming out usually around the middle of August through the beginning of September. Japanese maple scale, incredibly difficult to manage. Many different hosts. In Delaware, we often see the first generation start around 695 growing degree days, which is corresponding to late May, early June, depending on the given year. Infestations can be pretty high. Much like gloomy scale, the covers do not come off from year to year, so you have to be careful. If you do an application for this, you need to take a sample and look at it under a dissecting scope to see how efficacious your management efforts were. Maryland found growing degree days at 816 degree days with activity for crawlers that continued for 8 weeks. So it can be difficult to get this under control because of overlap of generations, which I try to illustrate here with a calendar representation down below. Distance and Taos are insect growth regulators that provide good to excellent control if applied at crawler activity. Port oil insecticidal soap applied at crawler activity also provide good to excellent control. Safari and Altus are 2 products that give excellent control. Safari is a neonicotinoid, Altus is not. One of the things you'll need to watch for as you look at a sample infested with Japanese maple scale, this white arrow is pointing to an evenly edged circular hole in their cover. That is evidence of parasitoid activity. There's a number of parasitoids that will attack Japanese maple scale and other armored scale in our area. The other thing you're looking for, it's a little harder to see in this picture, is crawler activity. You want to know if you've got crawlers active. So if you've got a sample that you treated last year, you want to look at it this year before you make treatments to see if you're going to be wasting time and money in product or not. Or if you need to make an additional follow-up application this year. So when we sit at the scope, we'll look, they generally are white and elongated or a little curved, but that can wear off and you will see what remains of the test, which is the brown section you see here. Here we have a crawler, they're purple in coloration. Now this is all in one sample, so we have a number, or the infestation looks high. I probably flipped over all of these, or this number, looking for pictures for this. I would say probably a third to almost half had nothing underneath it, meaning there was just nothing there to show. So I'm not showing you pictures of nothing. If we look close, we have a female that had been feeding underneath one of those covers. I'd flipped it over. Here's the test. Here she is. She's got a purplish coloration to her as well. Here you see another female, and here you see eggs. And in an earlier picture, we saw crawlers. There's a crawler right here. So it's a problem that you have crawlers, eggs, and females active all at the same time, because if you're using horde oil, insecticidal soap, or an insect growth regulator, you have a chance of missing a critical life stage for control because they're at a non-susceptible stage. Insect growth regulators are going to have a hard time having impacts on mature females because they're already mature. They're also going to have little impact on eggs because they're not moving around or feeding. They have their best efficacy on crawlers and early settling nymphs. Well, at any given stage, being present, you're going to have a stage that's not going to be targeted at any given time on your plant, which is why you need to go back and follow up to check on how well your products did from year to year when targeting armored scale. In particular, elongate hemlock scale, which is on a different group of plants, or Japanese maple scale like we see here. Here's a close-up of some of the eggs underneath one of the coverings. They're not feeding. It's hard to get control of the eggs because they're protected by the cover that the female had produced. She lays the eggs and she dies. The eggs remain until they hatch and then they crawl out. Here's examples of crawlers. Again, close-ups, and here you can almost see the eye spot. The camera I was using didn't have video capability, or at least Tracy and I didn't know how to utilize it to get to show video of these guys crawling around. It's also why they're not in focus. Here's I don't know how this fungus got in here. It's weird. But I think this is like called the dog vomit fungus, so I thought it'd be fun to mention. Here's a video of a potential beneficial. This could have been a spider, but as we'll see, there was nothing in there. But you will see spiders that will make nice little nests like that to wait for caterpillars to come by. There's nothing there under, unfortunately, in this instance. Let's see if I can get it to move. So the next slide. Okay, let's see. Come on. There we go. This next one is an image. So these oak leaf tires, or leaf tires, can be on oaks. This one happens to be on sweet gum. You can also get them on red buds. These do not require a management option. But sometimes samples like these will come in and they'll send in the grouping. Out in the field, it's just as simple as pulling it apart. And here you can see the caterpillar that had tied the leaves together. It does that to help provide a nice secure location to feed. The amount of damage they do is negligible. Let's see if I can get this to move. Yep, so I'm almost done, I promise. This is a cool video. You may find these are a ground-dwelling bee. They'll fly and swarm all over the place. I was moving rocks around them. They're non-aggressive, solitary, or communal, but not in a hive like a yellow jacket. They're non-aggressive. They're not likely to sting unless like you pick one up and try to pull its legs off. Then it would probably sting you. But generally speaking, they can be in your landscape and they're not going to cause any problems. We typically see these in late April through May flying around. This is a species that happens to be active later in the summer. We'll have a couple more, one that people are frequently familiar with, the cicada killer, also a big ass wasp that's non-aggressive and very unlikely to sting. I've dug up the nests with the female right there, and she was crawling on my legs and didn't sting me. We have another one. It's a scoliid wasp that also is active in the fall or late summer, early fall. That's non-aggressive, that will fly over turf. This is what I think most non-entomologists question is, why doesn't mosquitoes suck fat instead of blood? I think that's an awesome question. And at that, I guess I'm done.